Please welcome Matías Peire, GRIDX.
Okay, thank you again. For those who are not at the morning, my name is Matías Peire. I'm the founder of GRIDX, that is a venture capital fund in Latin America focused on very early stage for biotech startups. And today we are presenting our ArgenTAG, one of our portfolio companies that we have more than 50 portfolio companies in our portfolio. 70% of that founded by women, for you to be happy. And ArgenTAG is working in the convergence of biology and electronics and software, trying to get more tools for improve the single-cell sequencing technologies. So, welcome, Leandro.
Breaking the wall to single-cell genomics. Leandro Siapina, ArgenTAG.
Okay. So, imagine that you want to watch a movie. You sit on your sofa, you turn on the TV, and you only watch the last 10 minutes of the movie. You'll probably get an idea about the story, but there's still a huge amount of information that is still missing. So actually, this is what's happening today when researchers, biotechs, or pharmaceutical companies want to study the gene expression of the cells. They can only capture a small fraction of the information. I'm Leandro Siapina, co-founder and COO at ArgenTAG, and we have a solution for this. Cells are the basic unit of life, and in order to reveal the hidden mechanisms of critical human diseases, we need to understand what's going on with the cells. Not only the end of the information, but the complete information. So at ArgenTAG, we are developing a high-resolution kit to reveal the complete RNA landscape of the cells in a cost-effective and scalable way, so researchers, biotechs, pharmaceuticals can unlock new answers in basic research, drug discovery, or even cell therapy processes. So what we have is a proprietary barcoding technology that works over long-read DNA sequencers, even the portable DNA sequencers. Then we have a wet lab protocol that is able to tag each of the cells with a unique barcode in a robust way that can overcome the long-read sequencer noise. And then we have a software solution so researchers can visualize the results once the sequencing is done. During this year, we achieved 2 strategic milestones. On one side, we published our core barcoding technology in scientific reports, and we sequenced the highest ever number of barcodes for a long-read DNA sequencer. And best of all, we achieved high-quality results that are comparable to existing or established short-read sequencing technologies. So we have 80% read recovery rate, and then we have 0.2% of crosstalk. On the other hand, we were accelerated by IndieBio New York, and we did the first experiments with real cells. We mixed 100,000 cells, different types and different concentrations. We tagged and sequenced all of them together, and then we successfully recovered the complete profile, even the one on lower concentrations. So we are building our company based on a customer discovery process, and we are covering an unmet need in the market. This year, we signed 10 LOIs with genome centers, biotech companies, and PIs in the US, in Latin America, in Germany, and also in Australia. And now we are preparing 3 strategic collaborations in the US to kick off in 2023 with our seed round. And this will be towards collecting data about the single-cell kit, this high-resolution kit, and then achieving a commercial version for early 2024. This is us. We're a team of engineers, biotechnologists, molecular biologists, and business specialists. We have more than 20 years of experience pushing the limits of genomics. And we have a key now to democratize single-cell resolution, single-cell sequencing technologies, and unlock a $10 billion opportunity by 2030. So our goal is to be the leading company offering single-cell sequencing solutions on top of long-read DNA sequencers, and we are building our company for that. We have a great team of advisors. We have top biotech investors, And now we invite you to join our journey and watch the full movie of life. Thank you very much.
You have quite a few women in your team. And
Yeah,
we know that
more women than men.
Yes, and actually sustainability from economic sustainability, but also social sustainability and ecological sustainability is great. So let's go. Oh, that's a comment. Let's start with a comment.
Okay, great.
For a change. Oh, you meant a question?
The sign, yeah. So the technique is a hot topic and it's quite fascinating as you described it, but it's very pricey, as you said. And you said you would have the cost-effective solution.
Yeah.
First of all, how cost-effective are we talking compared to the commercial kit? Second of all, you said the commercialization will happen by 2024. Would you be open for collaboration, sending samples to other institutes for testing, for example, from now onwards, or shall we wait until you release it?
Yes, thank you. So we'll offer, our business model is to offer the single-cell sequencing kits. So we can discuss about sending a kit this year or early next year. So then you can give us feedback on that. On the other hand, you mentioned cost-effective. So today's solution, you need like the 10x Genomics, and that's a microfluidic that then you will complement with the Illumina sequencing, and that's very expensive, like the machine, but also the reagents and all of that you already know. So our solution is without microfluidics. So we have a kit, and it's like pipetting with a combinatorial approach, let's say, and then you put everything together into the DNA sequencer, that it could be a nanopore or a PacBio. In the case of a portable DNA sequencer, like it's a machine comparable to an iPhone, let's say, so you have our kit that could cost, in the best scenario, like a small-scale kit, $5,000, and then with a starter pack kit from Nanopore, like another $1,000, let's say. So it will be like 50 times cheaper than 10x, Illumina, and all the other solutions. Yep.
Mm-hmm. The next question from here, and we will have the next one here.
Thanks. You made that super intriguing movie analogy, but are you able to get temporal resolution, or are you just getting better resolution at the same point of time?
Yeah, so existing solutions run on top of short-read sequencers. That means that they can read small fragments of DNA. Now we are building a native solution for long-read DNA sequencers. So we can have more information, more biological value, and take better decisions on that. So that's why we say that today's solutions are low resolution, and that our solution that will work on long-read sequencers will be higher resolution. And actually, well, with these genome centers that we're speaking and with biotechs, they are very interested here because they're missing information here. Like isoforms, for example, they can capture with long reads, and they cannot capture with 10x Illumina with the short read solutions. So that's why we say that we can reveal the complete RNA landscape of the cells.
Mm-hmm. We have one question here and one last one here, please.
Okay.
Yeah, it's intriguing. You're saying cell. So what cell platforms can you, does it work for your technology? I'm in the back here.
Oh.
So yeah. So what cell which cell platforms are you currently, your solution works on?
What do you mean with cell platform?
Well, you know, you have mammalian cells and you have fungal cells and you have
mammalian
yeast.
cells.
But are you thinking of expanding it?
Now we did some tries in New York in biolabs with different types of cells, but we're focused on mammalian cells right now. And then we can discuss what you're interested in. Yep, thank you.
Thank you. We have one question here.
I got the impression that you were having very high accuracy, but I understand nanopore is about a 5% error. Did you say you're better than that with nanopore technology?
Yeah, actually, that's a great point. Our core barcoding technology and the publication that we did this year, it was about how to overcome the error rate of nanopore sequencing. So we can take this highly noisy instrumentation like the nanopore with 10% of error rates, and we can convert that into a, let's say, a highly accurate machine. And that's what we did, and we have like the same quality that Illumina sequencing, 80% read recovery rate and 0.2 crosstalk. So actually, we're now focusing on single cell, but we have a lot of room for other applications in the long-read sequencing space.
Thank you very much.
Yep.
That was
Thank you.
the word of the end.
Thank
you.
Okay.
Thank you.
Thank you.